Good morning, everybody. I'm Lori Burrows. I am the Associate Director of the IIDR, and it's my pleasure today to introduce our last IIDR round speakers for 2023 spring season, Dr. Shariq Haider and Yue Wang. So Dr. Haider is a professor of medicine at McMaster University. He serves a wide variety of leadership positions in infectious disease, including research lead for transplant infectious diseases at the Juravinski and medical director of HIV care in Hamilton. Today, he's going to be talking to us not about HIV, but about Zygomycosis care and the Canadian Mucorales Registry. That's a mouthful. Yue Wang, our second speaker, is a senior PhD student in the Zhu lab here at McMaster, where she focuses on population genomics analysis of several human fungal pathogens with an emphasis on Candida auris. Today, she'll be talking to us about her research, which is almost done. She told me she's working on her PhD thesis, which aims to investigate the natural habitats, transmission routes, evolutionary history, and antifungal resistant mechanisms of Candida auris. So thank you both for speaking today. I'm going to invite the audience to put any questions that they may have in the Q&A box, and we'll answer them at the end of the two talks. Okay, Dr. Haider, are you going first?
Yes, thanks very much.
I will pass it over to you then.
So a real pleasure, Lori. Thank you for that kind introduction. And it's a real pleasure to present with you. I'm really looking forward to her talk on Candida auris. So my topic is titled Zygomycosis, an emerging fungal pathogen. So the objectives of the next 20-25 minutes is to overview a little bit about the epidemiology and mainly about some of the deficits or gaps that we have around the epidemiology of mucormycosis or zygomycosis. We'll talk a little bit about diagnosis and really diagnostic challenges that exist, again, and treatment, mainly in the context of the updated zygomycosis guidelines. But I'm really looking forward to sharing with you some new data. It's a registry that I'm leading. It's a pan-Canadian registry of about 13 Canadian centers that's trying to address some of these gaps in epidemiology. It's titled the Canadian Mucormycosis Registry, and this will just be the interim data that's being presented, which we recently presented at the ECCMID meeting in Copenhagen. So I'll start off with a case just to highlight the burden of disease for mucormycosis and the complexity of this disease. So this is a 55-year-old Caucasian female with underlying myelofibrosis who's JAK2 positive, who underwent a bone marrow transplant. And this is, for those who don't know transplants as well, there are different types of transplant. This is called a haploidentical transplant. So her donor was in fact her brother. I mention these facts because being a haploidentical transplant, having underlying myelofibrosis and receiving a JAK2 inhibitor are all risk factors for invasive fungal infection. She also happened to be CMV positive and recipient positive, and that usually means that there will be multiple CMV reactivations post-transplant. And that's mentioned because CMV is also a risk factor for invasive fungal disease. This is sort of her, unfortunately, the reality of transplants. Often they're complex courses, and this is a sort of a timeline of what happened to her. So day one, after she got her cells, she developed something called CRS, cytokine release syndrome. It's not that unusual. She was treated with an anti-IL-6 agent called tocilizumab, which people are familiar with in the days of COVID now. And then she went on to receive GVHD prophylaxis, or what's known as graft versus host disease, which is one of the complications that one can see post-transplant with immune suppressants, MMF and tacrolimus. And then her clinical course gets complicated. On day 11, she develops fever and neutropenia and gets her standard antibiotic therapy called piperacillin tazobactam here, which is our standard regimen for fever and neutropenia. About 7 days later, she develops pleuritic chest pain and some hypoxia. And her CT, and I'll show you some pictures, show multiple ground glass subsolid nodules. When we see this appearance, the concern about invasive mold infection, particularly invasive aspergillus, becomes certainly very prominent. So this led then, this is the pictures. So you can see on the left, this sort of ground glass area that you're seeing on the picture here. This is sort of the largest one, but there are multiple subsolid and solid nodules that are being seen. So that led to a test called a bronchoalveolar lavage, where we took sampling from the lungs. Not unusual. The culture, she didn't grow anything. And this is one of the challenges of mold disease, is that the sensitivity even for aspergillus culture in the BAL is no higher than 30%. But we now have some newer diagnostic tests, and we used a non-invasive marker called galactomannan, which has greater sensitivity and specificity when you do it in the BAL. And it was strongly positive at 2.03. So the new cutoffs are, values above 0.8 as being positive, but this was strongly positive, and we had a diagnosis of invasive aspergillosis as early as 20 days post-transplant, not necessarily the best way to start off your transplant. So at that time, we were running a clinical trial which was a multi-center randomized control trial of voriconazole versus posaconazole for people with this disease of primary invasive, proven or probable invasive aspergillosis. So she went on the study, was on about 6 weeks of treatment when we did our first assessment, but unfortunately, based on that assessment, she had actually failed on the clinical trial. She had new pulmonary nodules that were being seen. So at that time, she was escalated to a combination therapy of posaconazole plus caspofungin, which is an echinocandin with a triazole. And she goes on this for another 30 days and has a response at that 4-week mark. At this point, she actually has a mixed response, and there is some stability by what we call the EORTC criteria. So we're pretty happy at about 4 weeks. That's about the best that you can express in this heavily immunosuppressed patient. So this is kind of what we're seeing later on. You're seeing a much larger nodule that you're seeing here, and you're seeing some areas of cavitation that are present as well. Now, the journey goes on. We're into day 273. So, you know, we've really extended this out, and we're coming to, you know, roughly 7 months, 6-7 months on treatment with posaconazole and caspofungin, but she was stable on treatment. When she developed, unfortunately, new symptoms again, new and worsening pleuritic chest pain. She has another CAT scan done at the time, which now unfortunately shows that there is further disease progression on this combination therapy. At this point, she gets a repeat bronchoalveolar lavage, and this time, even we do actually get successful on culture. We identify actually mucormycosis. And this is one of the points to make, is that one of the drivers of mucormycosis can often be selection pressures from prior azole treatment, particularly prophylaxis, but certainly even prolonged treatment. So she has no evidence at the time of GVHD, which is good news for her. At that point, the decision, because she now has rhizomucor, was to switch her to the drug called amphotericin, or ambisome, which is the lipid formulation of it, is what she got at a dose of 3 milligrams per kilogram per day. She's actually was doing quite well on the treatment for about 3 months, and then we see the usual complications of amphotericin come up. She developed renal impairment. And so usually what we do to try to manage this, because choices are limited for mucormycosis treatments, is that we adjusted her to 3 times a week amphotericin. And you can see from that prior picture and this current picture that we are, you know, there is stability that is being seen. And look at this. This is actually while on treatment with ambisome. This would still be regarded as a very reasonable response on treatment. So she was doing well from an efficacy perspective. The challenge, unfortunately, was the kidneys. So around day now 466, so this is now beyond a year of this unfortunate course of post-haploidentical transplant, we make a decision, because the kidneys are getting worse, to switch her to a new drug that became available back then in 2018. Now it's available for routine use. It's a new triazole called isavuconazole that has activity against both aspergillus and mucormycosis. The good news is that it does not have that renal toxicity concern. So she went on the isavuconazole, did really well on it, actually completed 4 months of therapy. But at that time, this was compassionate access, so we were only able to secure it for another sort of 3 months or so. And then unable to secure it. And she made a decision at that time to say she'd had enough. She was clinically stable. The lesion hadn't actually evolved on scans, and she was left for routine follow-up by her primary MRP team. Does the story end there? Unfortunately not. So then later in 2020, so this would be now almost, you know, I think we were here around 2018, I'll say about a year and a half later, she develops this left-sided abdominal pain and has this solid lesion that's identified in the left pole of the kidney. And when it first came up, everybody thought, which is not good news either, that this may be just maybe a cancer, renal cell CA. So she went for a biopsy, and lo and behold, in the biopsy, she did not have renal cell CA, but she had granulomatous inflammation with hyphae and spores, and these were non-septated hyphae, again suggestive that she had progression of mucormycosis. But remember, this is now off treatment for a year and a half. She's restarted on isavuconazole. And I'll stop the story here, but it just speaks to the complexity of these patients and this rising burden of mucormycosis in our hemato-oncology patients, particularly our hematology and stem cell transplant population. So a little bit about mucormycosis. It's an uncommon opportunistic fungal pathogen, but we do think that there's an underestimation of the burden of disease. The rarity of the disease has prevented sort of clinical trial study data, and the epidemiology to date has relied very much on cases, case series, and registries. And I'll share some of the registries that we have and then how we got to our particular registry. The diagnosis is challenging, and that we think leads to underestimation of the disease. But diagnosis is critical. I think this is a really important point, that in mucormycosis, survival is very much dependent upon early diagnosis. And it's based on a couple of observational studies that have been done, which have shown that the early initiation of amphotericin mucormycosis can change your mortality. So if you started at less than 6 days, your mortality is around 48%. If you have a delay of greater than 6 days, your mortality is 82%. Neither one is a good number, but absolutely knowing that this is mucormycosis and getting them on effective therapy impacts outcomes. And the challenge that we have is that often the clinical presentations, patient risk groups are often similar to primary aspergillosis as an example. So understanding the burden of disease in the developed countries, it's infrequent, but it's the second most common mold infection after invasive aspergillosis, and the highest risk groups are HSCT, hematological malignancies, and solid organ transplants. Well, but there's a complete dichotomous presentation in developing countries. It's a disease exclusively almost in patients with uncontrolled diabetes, but we're starting to see a surge as well in patients with trauma, and both developing and developed countries, you'll also see it in the context of burn patients. So understanding the burden of illness, again, I said again that there is challenges around incidence estimation and prevalence. One obvious challenge is that we're seeing an increasing susceptible population. There is a need for greater physician awareness, and I think these types of rounds are part of that physician awareness. There is a lack of standardized diagnostic strategies. We rely still a lot on imaging, but there are newer microbiological tools that we'll certainly talk a bit about. And then the other challenge is that there's no really harmonized denominators when you look at the existing limited data that's out there for cases, case series, or the limited registries. This is just meant to highlight this point that was shared in the case about the impact of prior azole therapy and the risk of invasive aspergillosis. This is a study just comparing patients who received mold active prophylaxis, and when they broke through, it was uncommon, but if you look at the top graph, it's the folks that broke through on prior azole prophylaxis. The first thing that there is that when they break through with not with friendly aspergillus, it's with Aspergillus usutis or Calidosis complex, which tends to often be very resistant to existing treatment options. And then the other thing that they often break through with is that 30% of them will break through with Mucorales species. So the selection pressure of azoles is certainly an issue. The bottom shows you the difference that they're not on azole prophylaxis and they get a non-breakthrough invasive mold infection. It tends to be primarily now invasive aspergillosis and tends to be the more common Aspergillus fumigatus as the pathogen. The next study I just quickly wanted to share with you is actually a paper. It's a really comprehensive paper actually by Dr. Prakash and colleagues published in the Journal of Fungi in 2019. I've just picked a couple of slides, but it highlights some important themes. This is a global epidemiology of mucormycosis based on, again, cases, case series, limited registries. And a couple of themes, these are busy slides, but I just want to highlight a couple of themes that when you look at the developing world context, we look at India and Iran, the single most important patient population is diabetes or diabetic ketoacidosis. But if I bring you over to France, if I bring you over to Europe, you start seeing that hematological malignancies and slowly solid organ transplants are becoming sort of the next group, but certainly that dichotomy is very well described in terms of risk groups. And how does that translate into the clinical presentation? Well, if we go to the developing world context in Iran or in India, because they primarily have diabetes as a major risk factor, the most common presentation in diabetics is always what we call ROCM, which stands for rhino-orbital cerebral mucormycosis. So the disease is mainly in the sinuses and in the brain. Whereas if you come to the developed world context of, let's say, in Europe here, you'll see that, first of all, we see a lot more hematological malignancy patients. And suddenly the presentation, we see some ROCM, but you start to see pulmonary disease as becoming the single most common presentation. And then, yes, there's differences absolutely in the genus and species, depending upon where you are. That's all this slide highlights, that if you go to India, looking at Rhizoba species, followed by Apophysomyces, which you don't see as commonly for sure in Canada or the United States or Europe. And then if we come to, say, France, we have Rhizoba species that are the most common, and then Leuthemia as being our second most common. So again, genus and species identification is important. So I mentioned that there is limited data out there. There's only really, there have been about 3 pan-fungal registries that I'm not sharing with you because they only had sporadic cases of mucormycosis. Most of those, the emphasis was on candidiasis. And those pan-fungal registries, it was actually, I just named one of them because it was a Canadian one done by Dr. Lindsay Nicole back in the early 1990s. And there were 3 Canadian centers, and Hamilton happened to be one of them. So we should mention that. But they were really focused very much on invasive candidiasis with sporadic cases of mucormycosis. And then we had the PATH registry that some of you may be familiar with. But to focus in on mucormycosis specific registry, there's really only two. So there's the retrozygo study. This was done in 2005, 2007 in France. They used French hospital information systems and National Reference Center for Mycosis and Antifungals. And they ended up with 101 patients. So in this disease, that's considered a lot of proven and probable mucormycosis. 60% of them were proven, 40% were probable. One really interesting observation of that study was the challenges in diagnostics, that only 50% of patients that were histopathology positive, that would have been the proven cases, were actually culture positive. So the limitations in sensitivity for culture was identified here. And then the second, which is probably the largest registry, is a collaboration out of Europe, published by Dr. Schiatta and colleagues from 1995 to 2001. This is actually a bit of a typo. It should say 13 countries, not 9 countries. So 13 countries put together in Europe came up with 230 patients. Now, their analysis, there was one important observation that they came up with, which is, again, they were able to validate that earlier observational data that I shared with you about the importance of early amphotericin use. They identified that both early surgery and early amphotericin on multivariate regression analysis was very important for clinical outcome and improvement in clinical outcome. So that's all we have. That's the existing registry data. And so quickly, a little bit about the challenges in diagnostics, because I've talked about that. So what are our diagnostic options? Well, you know, we start off, obviously, our gold standard reference standard is always tissue histopathology and direct microscopy. Challenges is that, you know, it is still a little bit of an art and a science. So sometimes being able to separate the septate and non-septate hyphae isn't as easy as one may think. And I know our microbiologists and our pathologists know that. Then we come to culture. The problem with culture is lack of sensitivity. So as I mentioned, only about 50% of histopathological cases will be culture positive. But at least with culture, you do get genus and species. And I once again emphasize the importance of the need to get to genus and species and to get to a diagnosis, because early diagnosis in this disease impacts outcome because of the need for early amphotericin use in the appropriate patient. So these are just some pictures showing that we still rely very much on radiology, and there are some radiological features of at least of pulmonary mucormycosis that's been very well described. The idea that if you have more than 10 nodules, it tends to lean more towards mucormycosis than aspergillosis. And then something called the reverse halo sign, and that's what these pictures here, my arrow is sort of showing you, which is this idea. The halo sign is what's described for aspergillosis, but reverse halo is this idea of a central hypoattenuation surrounded by a zone of hyperattenuation. So these are features that can increase your suspicion for there's something else called the vessel occlusion sign. And generally, if you have pleural effusions and you're suspecting mold disease, you should lean more towards mucormycosis than aspergillosis. But newer diagnostic methods is what we need, and that's where molecular methods have come in. And again, the ribosomal RNA locus is a common target for fungal identification. And the two, certainly the two areas that are most subscribed to is this area called the ITS, the internal transcribed spacer region. That's sort of the fungal DNA fingerprint for fungi, and is sort of the preferred target site for molecular identification. But in cases where that fails, the other site that has been certainly looked at by CDC, et cetera, is the 28S, which tends to have smaller amplicons, and certainly can be looked at in patients that do not have the fail to identify the ITS primer. So that moves us to this idea of just giving you this, I won't go through this in any detail, but to tell you that there have been updated guidelines for mucormycosis, and they reflect everything that I've just shared with you. But just to give you some general themes from these guidelines in 2019, is that the importance of imaging. So if you're dealing with a diabetic patient and you're worried about rhinoorbital cerebral mucormycosis, you must start with CNS imaging. If you're dealing with pulmonary mucormycosis, you should start with chest CT imaging. The importance of early sampling is again identified, and the three methods that we've discussed: direct microscopy, culture, and then where obviously when you're unsuccessful, the importance of looking at molecular identification. Surgical debridement is a key part. And as I mentioned, there have been some data in the past that suggested early surgical debridement does impact on improved outcomes. And in this disease, in these guidelines, you can see the green is the highly recommended choice, that amphotericin remains the drug of choice. So it may be renally toxic, but it is the primary drug of choice for disease, particularly for brain disease, where you should be using much higher doses of 10 milligrams per kilogram. But there are some newcomers and people with pre-existing renal disease. The drug that we used here called isavuconazole in our case, and then posaconazole that does have activity against invasive aspergillosis. So I did want to quickly just share one interesting diagnostic piece that was a study out of 2016, which was actually it's a quantitative serum PCR. And for those of us that practice this disease, we know that sometimes patients are so sick that we're unable to get BAL samples in them. So wherever you can get a serum-based testing, it's always of interest in this patient population. So this was a retrospective French study looking at a quantitative serum PCR. The assay was able to pick up what will be our three most common genus and species that we see here: Rhizomucor, Rhizopus, or Mucor, and Lichthemia. And they took serum from patients with proven and probable invasive mucormycosis. Most of these were stem cell transplant, hematological malignancy patients, 34, 2 SOTs, and 3 diabetics. Most had a mix of pulmonary or disseminated disease. And the quantitative PCR had a sensitivity of 81% and 92% when insufficient sera was excluded. So that's really good. But the most important piece was that this serum was positive 9 days earlier than what you saw in either culture or histopathology. And if you remember, I said earlier that early diagnosis in mucormycosis improves outcomes. So this would obviously be of some interest. Again, on the same serum quantitative PCR, earlier detection, again, may serve as a potential screening tool. We do, as an example, in invasive aspergillosis, now have serum galactomannan as a screening tool. So this is promising. Obviously, we need further data, further validation. And one could even say that this may be a potential tool for monitoring response to antifungal therapy because it is a quantitative PCR. Again, some little aspects about that quantitative PCR. The sera here had pretty high concentrations of DNA. Typically, for invasive aspergillosis, we're dealing with pretty low concentrations of DNA. And again, there is some suggestion that could correlate to late stage disease in study patients. So then I come to my last bit, which is really where we came in. We thought that there was a gap in mucormycosis. There's only 2 registries that are out there. I've shared them with you. And we put together this registry that I've been leading called CANMUS, which stands for the Canadian Mucormycosis Registry. And it's a pleasure to present on behalf of all these investigators. And as you can see, there's a very broad east to west representation across Canada. And this is just interim data that we're sharing with you. So this is a 10-year retrospective registry. First of all, it is the most contemporary registry because all the prior registries ended at 2010. We started calculating data from 2009 to 2020. The goal was to capture the burden of disease, risk factors, clinical presentations, diagnostic strategies. So a lot of qualitative data around that. We use microbiology and pathology records at each site. In Quebec, we had a central public health lab where we were able to access data as well. Data was entered into REDCap. And the outcomes of interest were 6 and 12-week survival data, as well as global clinical response at 6 weeks, 12 weeks, 6 months, and last clinical follow-up based on a standardized definition called the EORTC definition of response. Our analysis that I'll be sharing today will be some descriptive statistics for both normally and non-normally distributed data. I'll be sharing with you some interim survival analysis that we presented at ACMID. I unfortunately will not be sharing some of our, obviously, our analysis. We're looking at risk factors and binary outcomes. That data is still being processed, but obviously of great interest. So this is the data as it's shared. First of all, these are not mutually exclusive categories, which is why you'll see that numbers don't add up to 100%. But the important point here is that in our 14-center experience, and there are still 3 centers that have not entered data into this registry yet, so about 11 centers, that you see the shift that's happening. That certainly in the context of the developed world context, we're seeing mainly bone marrow transplant and hematological malignancy as being very important. And in the hematologic malignancy, the single most important malignancy was AML. Yes, we still are seeing diabetics, but certainly less common than hematological patients. But 2 other really important factors is, again, steroids. And this is the one important recognition that's not been shown before, is the role of immunotherapeutic and molecular targeted agents. So the world of acute myelogenous leukemia therapy really changed in the last 10 years. Prior to 10 years ago, we had one standard treatment for everybody. They all went through standard induction consolidation, but now we use molecular targeted therapy. So that's a really important observation out of this registry. Sites of infection, again, is showing you the shift that's happening in the developed world context, that though we see that ROCM, rhino-orbital sinus disease, the most common presentation is pulmonary disease, as you can see here. And most of these, again, were proven in probable cases. I emphasized the importance of prior azole prophylaxis and the impact of that on the risk of invasive mucormycosis. So we did see that here as well. So 65% of the patients had broken through on prophylaxis. About 35% had been on some form of targeted therapy when they developed mucormycosis. The other interesting observation in this 11-center experience so far, this is interim data, and we're up to about 110. It says 108 of the 110 patients, so this would now be the second largest registry already globally, is that we're seeing mucor as being the most common species, followed by rhizopus, which is a shift in what's been seen previously. I see a little bit of rhizomucor and lipemia. Again, the importance of antifungal therapy is obviously been picked up, and we'll be getting into much more granular data in that as time goes on. In terms of response is always looked at as a composite of complete and partial response. And if you look at it at 6 weeks, we're looking at about a 30% response, which is about what you see for this type of disease. So not out of keeping, but you see the significantly high mortality rate of exceeding 30% at 6 weeks in this disease state. So this is just the survival analysis that was carried out, just to show you again. So roughly it's just 6 and 12 weeks survival, about the same, 67% survival at 6, 66% at 12 weeks. But we did do a survival analysis and came up with some important observations that, again, bone marrow transplants are absolutely an important driver of mortality. So you see the hazard ratio is 2.24 and the survival drops to around 45% in bone marrow transplant patients. The second really important observation, which has not previously been shown in any prior literature, is this idea of the importance of molecular targeted therapy as an independent driver of mortality. And so we're going to obviously have to look into that with some detail, because this is a mixed group. And so it's, people want to know what molecular targeted therapy. Are we talking about JAK2 inhibitors? Are we talking about our BCR-ABL inhibitors, tyrosine kinase inhibitors? And so we'll be looking into that. This is just preliminary data that's been shared. This is just a last slide, just to show that obviously other factors that have been looked at previously were looked at in terms of the survival analysis. And just the only one I want to highlight is, again, that yes, amphotericin absolutely was a predictor of improved outcomes in survival. So that's been shown in previous studies and again has been emphasized here. So I will stop there and turn it over to you for her presentation. Happy to take some questions at the end.
Thank you, Shariq. We're going to hold the questions until the end, for time's sake. So I will now invite you to share your slides. Thanks.
Okay, thanks, Laurie. Thanks, Dr. Shariq. That was a great talk. So now I'll share my screen. Okay, good morning, everyone. Today I will share our findings, including natural reservoir of Candida auris, antifungal resistance in our samples, and the genomic variation affecting antifungal susceptibility. So as some of you may know, Candida auris is a species fungus that can cause severe infections in humans. This fungus was first reported in 2009 and has since caused the nosocomial infection in over 50 countries. In October 2022, the World Health Organization classified Candida auris as a critical human fungal pathogen. Here are several characteristics that make this species one of the great global health concern. Firstly, Candida auris is capable of causing both skin and blood infections. And the people with blood infected by this fungus usually have a high mortality rate, ranging from 30 to 60%. Secondly, this species is quite resistant to multiple antifungals. According to the data from the United States in 2018, 90% of clinical isolates were resistant to fluconazole, while 30% also showed resistance to amphotericin B. Around 5% were resistant to echinocandin. Thirdly, Candida auris can tolerate a lot of stresses, including high temperatures and high salinity conditions. Lastly, certain strains of Candida auris can persist on surfaces for at least 2 weeks, making it really hard to get rid of them from clinical setting. So based on the genomic analysis, clinical strains of this species have been classified into 5 distinct clades exhibiting certain phylogeographic patterns. Specifically, clade 1 strains were predominantly isolated from South Asia. Clade 2 were originated from East Asia, while clade 3 strains were mostly reported from Africa, and the clade 4 strains were isolated from America. Currently, there are only 5 or 7 isolates of clade 5 strains, and all of them were isolated from Iran. Since people travel to other countries for medical purposes, Strains of Candida auris from multiple clades have been reported from the same geographic areas as reported by a lot of studies. Indeed, okay. So here are our strategy to combat the fungal burden. So antifungal drugs are prescribed to treat fungal infections. Commonly used antifungal drugs include azoles, ponins, and echinocandins, and each with its own unique mode of action. Those interrupt the ergosterol biosynthetic pathway, a key component of fungal cell membrane, while ponins bind direct to ergosterol and form channels through the membrane, disrupting its integrity. Echinocandins, on the other hand, inhibit beta-1,3-D-glucan synthase, an enzyme that is necessary for the synthesis of a key component in the fungal cell wall. Indeed, the extensive use of antifungal agents can lead to the development of antifungal resistance, presenting an additional challenge in combating fungal burden. So my presentation will focus on two sections. The first section is exploring potential ecological niches of Candida auris and antifungal resistance patterns in our sample. Before 2020, Candida auris was only isolated from clinical environments. So this arrests the question of its origin, whether Candida auris originates solely from clinical environment, or if it had sources in other natural environments. Besides, species close related to Candida auris have been identified in various ecological niches, such as soil, fruit, and animal samples. Therefore, we conducted 3 collaborative studies with Dr. Anu Chaudhary from the University of Delhi. In those studies, we investigated the tropical wetlands, fruit samples, and dog samples as potential reservoir for Candida auris. So the first study is environmental isolation of Candida auris from coastal wetland of Andaman Islands, India. The Andaman Islands are a group of isolated tropical islands in the Indian Ocean. 8 sampling sites were selected based on the geographic heterogeneity and the ease of accessibility. They include salt marsh, tourist spot, rocky shore, and the mangrove swamps. For each of the 8 sampling sites, we collected 3 replicates of seawater samples and salmon samples. Those samples were plated on SD agar first, and then the colonies were transferred to Chrome agar for yeast identification. Then those yeast species were further confirmed using method of mass spectrometry and the ITS sequencing technique. So various yeast species were isolated from the 8 sampling sites. But we didn't identify any isolates, any yeast species from site F. Candida auris was only recovered from 2 sites, site A and site B. Site A is a salt marsh wetland characterized by a lot of seagrasses and seabirds without human activity. Site B is a tourist spot. So where 22 isolates were collected from this site. Then we conducted a whole genome sequencing analysis on selective strands, and then antifungal susceptibility test on those samples. The file entry on the right side revealed the relatedness of 13 wetland isolates and the previously clinical strands collected from India. All the wetland isolates with their label highlighted orange. The color bar indicate their sampling location. 2 of them from site A and 11 of them from site B. All of the wetland isolates belong to clade 1, consistent with other clinical isolates collected from India. Of note, 12 of them form a clonal cluster on the phylogenetic tree, but one strain 176 located somewhere else on the phylogeny. So here shows the MIC distribution of several tested antifungals, including 5 azole drugs, fluconazole, itraconazole, voriconazole, isoconazole, and posaconazole, aflibercept B, and 2 echinocandin drugs. So only the Only one strain showed resistance to all tested antifungals, the strain 176. All other 23 samples were quite resistant to all tested azole drugs and eventurin B, but none of those strains were resistant to echinocandins. So the second study is Candida auris on apples, diversity and clinical significance. In this study, 84 fruit samples representing 9 fruit species were collected. We screened the surface swabs and the internal tissue of each fruit sample for Candida auris. We observed various yeast species on the surface of those fruit samples, but Candida auris was exclusively isolated from apples. So in total, we collected 62 apple samples, including 2 apple variants, Red Delicious and Real Gala. These apples included 20 freshly picked apples from orchards and 42 stored apples from local markets. Among the 62 apples, 8 yield Candida auris. A total of 16 Candida auris colonies were recovered from 8 apples, and all of the 8 apples were stored apples. The fungal tree revealed the genetic relatedness within the fruit isolates and their relationship to clinical isolates from India. Different apples were indicated by different different color apple figures on the fungal tree. The 16 apple isolates can be classified into 5 clusters, A, B, C, D, and E. Here are some interesting observations. We found genetically similar strains isolated on different apples as shown in cluster B and C. But strains with different genotype can also be found on a single apple, as indicated by the red apple, where different isolates belong to clade 1 as well as clade C, while the blue apple contains strains belong to clade C as well as E. Of note, Candida auris strains on surface of apples are quite related to clinical strains. In clade 1, in cluster A and D, fruit isolates different from the clinical isolates by only several SNPs across the whole genome, suggesting their shared ancestry. So then we compare the antifungal susceptibilities of medical antifungals and the agricultural fungicides of the 16 fruit isolates with clinical resistant strains and clinical susceptible strains. We found both apple isolates and the clinical resistant strains show resistance to fluconazole and triazole fungicides. But the apple isolates also exhibit elevated MICs against other tested azole drugs, including voriconazole, itraconazole, isoconazole, and posaconazole. So now let's look at the third study, Candida auris in dog ears. In this study, we sampled skin and ear swabs from 52 stray dogs that were admitted in the ICU with severe skin infections and certified pet dogs that visited vet clinics. Hands of healthcare workers in clinics were also examined. Then we conducted whole genome sequencing analysis on recovered Candida auris strains from those dogs. In addition, we performed metabarcoding analysis on ear swabs of 10 dogs with Auris externa to look for Candida auris. So here are the culture-based isolation results. So overall, 2 of the 87 dogs contain Candida auris in their ears. Dog 1, D1 dog had a skin infection as well as otitis externa. 2 isolates were collected from this dog with a 1-month interval between them. This dog was treated with azole drug. Dog 4 only had skin infections, but Candida auris was also recovered from its ear. 4 strains were isolated from dog 4, from D4 in total. Both dogs were resistant to fluconazole, but the dog treated with azole drugs showed a much higher MIC to fluconazole. We construct a phylogenetic tree, including dog ear isolates and K1 clinical strands from various geographic locations, with each location represented by a distinct color on the color bar. And the different triangles represent the various isolation sources of Indian strain. The two strands from dog 1 are closely related to each other, as well as other clinical strands isolated from both India and other countries. But the 4 strands from dog D4 were very different and only similar to the reference strain. Here shows the metabarcoding analysis. We blasted the reads of each sample to the fungal ITS database to identify the presence of different species or genera and look for Candida auris. The heat map displays the read abundance of 28 genera across individual samples, and the 28 genera covered an average of 98% of the reads that were mapped to the fungal ITS database. The column name represent the sample name, while the row names correspond to the genus name, as well as Candida auris. As a result, we identified 3 samples showing sequence matching to Candida auris ITS region with over 94 identities. So to conclude, we identified tropical wetlands, surface of stored apples and dog ears as the natural reservoir of Candida auris. And all of our samples belong to South Asia clade, consistent with other Indian strains. In addition, we observed elevated levels of antifungal resistance in stored apples and dog ears, indicate that the application of antifungals could be a significant selective force for drug resistance in clinics. Lastly, the identified reservoir can act as potential source for the transmission of this yeast to humans and vice versa. In the second section of my presentation, I will present you the genome-wide association analysis of antifungal drug susceptibility in clade 1 of Candida auris. The rationales of these studies are twofold. Firstly, antifungal resistance is a great challenge to fungal treatment. Secondly, while many studies have described the mutations in antifungal-related genes of Candida auris isolates, but they didn't conduct a statistical analysis to assess their association with drug resistance in their samples. In this study, I collected 563 clade 1 Candida auris genome and conducted whole genome sequencing on those samples to identify single nuclear polymorphisms in those samples. Then I gathered the antifungal susceptibility profiles for 7 antifungal drugs. For each drug, I was able to collect MIC data for around 120 samples. Then the keywords analysis were conducted using Blink or Farm CPU to identify SNP associated with susceptibility against the individual antifungal drugs. So this table shows the significant SNPs identified linked to 4 other drugs. 2 of them introduced missense mutation that were highlighted in red, and 3 of them introduced synonymous mutation. The rest of them were located in indigenic region. Currently, no study has demonstrated the association between those mutations and antifungal resistance. Therefore, additional research or studies are needed to confirm this result. So this table shows the significant SNPs linked to echinocandins and aflatoxin B. A total of 2 missense mutations and 3 synonymous mutations have been discovered associated with echinocandin susceptibility. Of note, 1 missense mutation that converts serum to phenylalanine in the gene FKS1 has been proven to confer a moderate in vivo resistance against the echinocandins using a murine model of infection. Regarding aflatoxin B, 1 missense mutation and 2 synonymous mutations have been identified, but their roles in drug resistance need to be further confirmed through experiment. So in this study, we identified 2 missense mutations associated with drug susceptibility, 2 with echinocandin susceptibility, and 1 with aflatoxin B susceptibility. Our finding that the serine-2 phenylalanine mutation in FKS1 gene is associated with susceptibility to echinocandins, consistent with previous study. Other genes harboring missense mutation have not been annotated, so their roles in drug resistance remain to be explored. So I want to summarize my presentation with several key take-home messages. Our study have identified several previous unknown ecological niches of Candida auris, emphasizing the need for continuing exploration of its potential reservoirs. We also observed a close genetic relationship between environmental and clinical isolates, indicating the potential for transmission between different ecological niches. Additionally, antifungal resistance is quite prevalent in our samples, likely attributed to the selective pressure exerted by antifungal agents. Moving to the GWAS analysis, we identified several novel genomic variations associated with antifungal susceptibility, which can serve as diagnostic markers for antifungal resistance in clinics. Meanwhile, further research is required to gain a deeper understanding of the mechanisms underlying the antifungal resistance in this species. I would like to express my appreciation to my supervisory committee, my lab members, and our collaborators for their support and help. Thank you all for your attention. I'm open to any question.
Thank you very much, Yue. Appreciate both of your talks. They were great. Okay, so we'll start with some questions. Thank you. There's a question in the chat for Shariq. Very educative presentation. We saw a spate of RCOM in India during COVID, unlike other parts of the world. What could be the reason for this difference in the presentation?
Yeah, so you're absolutely correct. There was absolutely a surge, mainly in India, actually, and some other South Asian countries. Iran also reported a number of cases of mucormycosis post-COVID. Well, I think probably, the risk factor related, the association of diabetes with mucormycosis is all related to the issue that when patients develop uncontrolled diabetes, they develop an acidic environment, and that acidic environment, as you know, leads to liberation of iron from stored ferritin. And it's that iron that's the medium for hyphal growth. So I suspect that the association really is just when these patients got sick, they ended up with worsening diabetes, worsening DKA, and there was the association. And obviously we do better in the developed world context. People always wonder, why don't we see the signal here? And I think it's just got to do with better diabetes control, greater options, all that has helped.
I actually had a related question about this link with diabetes. So is there any information in the cases about, did this start as damage to your extremities and it's just that you're diabetic and so your blood sugar iron is higher? Because I mean, there's way more diabetic patients in the world than people undergoing transplant or malignancies, right? But it doesn't seem to be as common as you would expect necessarily.
No, absolutely. You're absolutely correct. Diabetes is a global disease burden that we see everywhere. And so the question of the chicken or the egg is always there. I think the other, link though, I think has to, again, I should just make that point, the other association between why we see diabetes, DKA as a risk factor of invasive mucormycosis. The iron is a big piece. The other bit, has got to do with this whole issue of neutrophil function and chemotaxis that you see an impairment as you get sugars above 12. So whether it's, again, the fact that, is it the primary diabetic insult that leads to this? Hard to know. I don't think it's been teased out, Lori, to be on that point. But I think why we see a discrepancy between the developing and developed world context, I think is entirely got to do just with better glycemic control here.
Cool. And I have a question for you. Is there anything unique about Candida auris versus other Candida species that we could use as a drug target for this very challenging organism? So something about its metabolism, or I know it's more heat resistant, for example.
Yeah, so that's a very good question. So currently, based on the GWAS analysis, I think the novel SNPs that I identified can be used to target Candida auris, right? Because they haven't been identified, haven't been documented in other species, in other related species. but currently we only use homologous genes to infer antifungal resistance in Candida auris. So that's the limitation of the GWAS analysis. But the novel SNPs could definitely be used as some novel drug targets or diagnostic markers for the for following studies.
And I seem to remember that when Candida auris first appeared, it sort of appeared simultaneously from multiple regions of the world. So is there any more insight into, how did we not see it 20 years ago, yet suddenly we're seeing it everywhere?
Yeah, so the reason, the Candida auris has very unique characteristics. They can persist in clinical settings for quite a long time than other close related species. So that's why it's hard to get rid of them and from clinical settings. And people travel around different areas for medical purpose, usually bring those Candida auris among different geographic areas. So that's the challenge for people to combat Candida auris. But yes, you're correct. Candida auris were simultaneous arrests at multiple areas and with different genetic background. So yeah, that's very interesting.
It's a mystery.
Yeah, that's the mystery.
Yeah, I think we're going to unfortunately be hearing a lot more about fungal infections as the climate warms, because we've been protected by our body temperature, but as they acclimatize to this, it's kind of a scary thought. So I'm hoping that those of you out there who are interested in developing antifungals will get right on that. Okay, I don't see any more questions, so it just remains for me to thank you both again for your great presentations, give you some applause, and thank our audience for their participation today, and we'll be back again in the fall. Have a great day.
Thanks very much, Laurie. Great presentation.
Thank you very much.